When doing research, it's much easier to work with small things than big things. So a lot of the research that contributed to the life cycle of a virus came from researching bacteria, not eukaryotic cells. Because we are talking about viruses infecting bacteria, these viruses are known as bacteriophages. If a bacteriophage kills the bacteria, it is known as virulent. If a bacteriophage enters the bacteria but doesn't kill it, it is known as temperate. For the lytic life cycle, the virus enters the cell, does some stuff, and kills the cell, and more virus spreads. It actually happens in 6 steps. First, the bacteriophage or the virus attaches to the host or the bacterium. Second, the DNA or the genetic material from the bacteriophage enters the bacteria. Second, the genetic material from the virus enters the bacteria by penetrating its membrane. Third, the DNA or RNA that was already in the bacteria is going to degrade, which leaves the only DNA there being the DNA for the virus. 4, the DNA from the bacteriophage is replicated inside of the host cell using the proteins from the host cell. The host cell also makes any other proteins that were needed for the virus to survive. 5th, all of those pieces of the virus reassemble inside of the host cell and get ready to leave. And 6th, the new virus actually leaves the host cell, and when it does, it breaks the cell membrane of the host, therefore killing or lysing the cell. So the lytic life cycle always ends with the host cell being lysed or killed. However, some viruses don't stay in the lytic cycle, especially if they know they don't have the environment for them to be able to just kill off a whole bunch of hosts. Sometimes they might choose that it's more beneficial to just stay within the host and let the host replicate itself, and eventually they'll come back as viruses, but we're just gonna stay as the DNA for a little while and stick around with the host. So when the virus or a bacteriophage enters a bacterium, and instead of deteriorating all of the genetic material from the bacteria, it incorporates its own material, and then instead of replicating the proteins and the other stuff needed, it just replicates its DNA when the bacteria replicates its own DNA. That is known as a prophage. Phage, again, being the thing that was going to eat the host cell, but it is not an actual bacteriophage. It's not actually eating the bacterium. It is before the ability to eat, or pro the phage period. Prophages go through prophase. Prophase being one of the stages of the cell cycle. So in the lysogenic phase, instead of the virus creating a capsid and lysing the cell upon exit, the virus inserts its DNA into the DNA of the bacteria and replicates as prophages every time the bacteria itself goes through prophase and the other steps of the cell cycle. Lysogenic leaves as prophage. Lytic leaves as bacteriophage. A lysogenic virus can re-enter the lytic cycle from different triggers. Some of those being the environment, so low pH or high pH, as well as just how many other bacteria are around. It can decide if it needs to make a bacteriophage or stay as a prophage. Some bacteria don't have the ability to kill you on their own. They're not pathogenic. They adapt or acquire a pathogenic aspect via a bacteriophage putting a plasmid into the bacteria. So the bacteria itself wasn't going to kill you, but now that there's this plasmid, every time that plasmid gets replicated or those proteins from the plasmid get made, the plasmid proteins will kill you. One example of this is cholera. The bacterium that causes cholera, Vibrio cholerae, is not actually infectious. What is infectious is a very specific plasmid that came from a different virus, aka a bacteriophage, and is now present inside of the bacteria. When a virus puts its DNA into a bacteria and changes the bacteria itself, that is called transduction of the bacteria. And that is one way in which bacteria can increase their genetic diversity. In summary, because it's easier to do research on small things than big things, most research on the life cycles of viruses has been done on bacteria, which means we refer to these viruses as bacteriophages, or viruses that are specifically infecting bacteria. There's two cycles that the bacteria can go through, the lytic and the lysogenic cycle. The lytic cycle is where the virus directly infects the bacterium. The bacterium degrades its own DNA, replicates everything needed for the virus, and the virus leaves, lysing the cell in the process. The lysogenic cycle, if a virus chooses to not make more of its own proteins and instead puts its DNA into the bacteria's DNA, it can replicate just to the DNA as something called a prophage. That prophage can become another bacteriophage if it creates more proteins and leaves the bacterial chromosome itself. A lysogenic virus can re-enter the lytic cycle by environmental triggers. Some bacteria are not in and of themselves infectious. They adopt infectivity via transduction by bacteriophages, or when the DNA from a virus is inserted into the bacteria via a bacteriophage.